A very good morning to all. This is the video demo lecture by Mugdha Karve from Department of Microbiology, SICS Degree College, Ambernath. This video lecture is regarding with the introduction to the enzymes. The enzymes are nothing but a very important factors. Those are involved in a catalysis of biological reactions or biological chemical reactions. So somehow they resembles to the catalysts which are present in or which we used normally for the activation or for carrying out the chemical reactions in chemistry lab. Okay, so for chemical reactions we used multiple types of catalyst so that the reaction will complete on faster rate. Similarly, in case of the biological systems like cells, the enzymes can act as a catalyst. We all know that any biological system, though it is of higher organism or of microorganisms, all are made up of cells. Every cell consists of multiple molecules, and all molecules are anytime they involve in some kind of reactions, though they maybe act as a substrate of the pathway or maybe act as an intermediate of that particular pathway. So multiple pathways like glycolysis, TCA cycles are carried out in a same cell at any given time. Okay, so the specificity or the separation of all these reactions or pathways, those are carried out by the means of specific enzymes. So every reaction is catalyzed by a specific enzyme. Thus, the enzymes are called as biocatalysts. So in introduction, we have to see that some kinds of general properties along with some historical aspects of the enzymes. The enzymes are first catalyzed or they are first invented by the scientist Payen with the discovery of enzyme diastase in 1833. As we all know that the enzymes are carrying out or they act as a catalyst for a biological system, so they are called as biocatalysts. The word biocatalyst is formed by two different words. One is bio, second one is catalysts. So bio stands for a biological system and catalyst stands for the factor that is involved to increase the rate of reaction. Most of the enzymes are proteins, whereas some RNAs also shows the catalytic activities, and such RNAs are called as ribozymes. Now the substance or the molecules on which the enzyme is going to bind is called as substrate. So the substrate is nothing but a specific molecule to which the enzyme is going to bind to form the enzyme substrate complex. The enzyme is further converted to the physically and chemically different molecule called as or the substrate is further converted to the physically and chemically different molecule called as product. So the enzymes is nothing but a factor that converts the substrate to their respective products. Next are some general properties of enzymes. A very first and important property of enzymes is specificity. As we all know that the enzymes are proteins, or most of the enzymes are proteins, so they are having some unique kind of amino acids or they are made up of amino acids. So specific amino acids are present at their active site. Active site is nothing but the site which is going to bind with a substrate. This specificity lies in what kind of amino acid sequence is present at the particular active site of enzyme. Okay, so the specificity is very important in case of catalyzing a specific reaction of a pathway. Thus, a specific enzyme is involved in a specific reaction of any kind of a pathway. So, if any pathway contains 10 different reactions or 10 sequence of 10 different reactions, then there will be the involvement of 10 different enzymes. So, every enzyme carried out a different reaction of that pathway. Now, the next property is their unique 3D structures. Again, the same thing, that is the specificity of the enzyme is lies in the conformation or the shape of a catalytic site. So nothing but this shape is maintained by a 3D structure of an enzyme. As we all know that the enzymes are proteins, so the proteins are having a specific folding pattern, specific 3D structure called as tertiary or quaternary structures. Okay, in these 3D structures or tertiary or quaternary structures, they act as an enzyme. Enzymes increase the rate of reaction by lowering its activation energy. Now, what is the activation energy is? As we know that the reactants are converted to the products. Now, during this conversion, the reactants gain some kind of energy or they required some kind of energy to convert itself to the transition state. In the given graph, the peak indicates the transition state of the reactants, so that this transition state is further converted to the product. Now, the conversion of the reactants to its transition state requires the energy, some kind of energy, and here it is denoted by EA, indicating that activation energy. Now, what enzymes are involved in? The enzymes involved in the lowering of this activation energy. Okay, so if this graph or we consider this graph, how we can say that this peak is lower down. Okay, so the reactants are immediately converted to their transition state and they are immediately converted to the products. So ultimately the rate or the speed of the reaction increases. The next property is enzymes does not consume in a chemical reaction. So what it means that whatever quantity of enzyme we initially provide in the reaction, we will get the same quantity at the end of the reaction, so that no enzyme molecule will be consumed in the reaction. Enzyme does not alter the equilibrium of the reaction. So in the same graph, we can consider that the location of that particular peak on the x-axis will never be changed, neither on left side nor on right side. Okay, so what is going to be happen is just the peak is lower down, but never it is shifted on either right or a left side, indicating that the enzymes does not change the equilibrium of the reaction. Now what is the equilibrium is? It is nothing but the presence of or the probability of 50-50% probability, that is conversion of substrate to product. Okay, so it is randomly, we can say it is an equilibrium. So most of the time we can say that this peak we observed at the middle of the reaction, so it is called as the equilibrium. The enzyme activity, most of the times it is affected by other molecules like inhibitors and activators. It is also affected by some kind of physical factors like temperature and pH. Here it is a mistake, that is at the pressure we have to consider that is pH. Okay, so for every enzyme there is optimum pH and optimum temperature. If the pH or a temperature is increased or decreased from its optimum quantity or optimum value, we consider the change in enzyme activity. This is all about the general properties of an enzymes. We just consider the reaction in which the enzyme is involved. Okay, so here it is a schematic representation of enzymatic reaction, that is substrate in the presence of enzyme, it is converted to the product. Remember, the thing is, the chemical and physical properties of substrate and product must be differ from one another, and this change is carried out just because of the specificity or affinity between the substrate and the enzymes, particularly the active site of the enzyme. This is all about the introduction of enzymes. Thank you.